Hi everyone, and welcome to our video about the light independent stage of photosynthesis. What we're going to be doing in this video then is having a look at two of the statements on your specification. 5.2.1e, which is looking at the fixation of carbon dioxide and the light independent stage of photosynthesis, and then 5.2.1f, the uses of triose phosphate. First thing then is we need to consider where this whole thing happens. So in our previous video we looked at the light dependent stage, and what we're going to be doing now is stage 2, the light independent stage. When it comes to the light independent stage, this is taking place inside the stroma of our chloroplasts. And if you remember, our little diagram at the bottom here should be a little hint for us as to where that actually is in our structure of the chloroplast reminder. We do have that outer membrane, the inner membrane with intermembrane space between them. Then we've got those stacks of thylakoids to make the grana connected by those lamellae. And then all around them, that fluid that we've got between them, that is the stroma. Now, what we find is in the light independent stage, it's light independent because it's not using light directly. That isn't to say it's not affected by light, because it does use the products from the light dependent stage. So without light, it couldn't happen, but it doesn't use light itself directly. So hence light independent. So before we really dive into the nitty-gritty of this actual part of the process, we need to remind ourselves about the other bit of photosynthesis that's going to be involved here. So in the light dependent stage, we had a look to see what happened to the water and how we made the oxygen. What we're going to be doing in the light independent stage is looking at the carbon dioxide. Now, the key cells involved here will be our palisade cells, which hopefully we remember a type of specialised plant cell that contain large numbers of chloroplasts. Now, those palisade cells are going to receive the carbon dioxide, which enters through the stomata, those little pores, and it then diffuses through the spongy mesophyll until it reaches our palisade cells. So that's one way that we get carbon dioxide there. The second is that because we have respiration taking place, one of those products of respiration is carbon dioxide, and therefore we can also utilise that source of carbon dioxide. So two main sources, either diffusing through the stomata, through the spongy mesophyll to the palisade cells, or as a product of respiration. What we're going to focus on then in this part of photosynthesis is something called the Calvin cycle. Now, this is named after the scientist that actually carried out the research into this, called Melvin Calvin. It's a great name for a scientist. And he actually was looking to see what happens to this carbon dioxide. So what we actually have then is carbon dioxide is inorganic. Now, what we do know is that once we start off with that inorganic carbon dioxide, we're then going to create an organic molecule, and that happens through this process of fixation. Now, unfortunately, that's not all we need to know as far as the Calvin cycle goes. As lovely as it would be to say A-level biology, all we need to know is inorganic carbon dioxide becomes an organic molecule through the process of fixation. It's a little bit simplistic, so we're going to delve a little bit deeper. Okay, let's dive into this then, the Calvin cycle in detail. What we will do is each little bit, take it one little baby step at a time, and then at the end we're going to tie it all together and see the whole cycle, because you know what, it's a cycle. It's called the Calvin cycle for a reason. Now we're going to start off then with our carbon dioxide. And the carbon dioxide is going to join with a chemical called ribulose bisphosphate. That S is important, folks. Now, we can summarise ribulose bisphosphate down to just RUBP. So RUBP, absolutely acceptable as the name for this chemical. You don't have to write the full thing. In fact, if you are trying to write it, a lot of people mess it up by missing out the S and therefore wrong. So RUBP, easiest way to do it. Now, in order to actually get the carbon dioxide combined with the ribulose bisphosphate, we need an enzyme, and that enzyme is called Rubisco. Again, the summary. Go with the summary, don't go with the full name, because the full name of Rubisco is ribulose bisphosphate carboxylase oxygenase. You really want to write that in your A-level exam? Because I don't think so. Let's just call it Rubisco, folks. Okay. Now, Rubisco is the enzyme that's then going to catalyse that reaction between RuBP and our carbon dioxide. So what we actually see happening, we start off with our RuBP, which is a 5-carbon compound, and then what's going to happen is our carbon dioxide, which is a 1-carbon compound, is going to join with it through the action of our Rubisco, and we're then going to form a 6-carbon molecule. Now, we don't need to know the name of that 6-carbon molecule, just that it gets converted. Now, both the RuBP and Rubisco are found in the stroma of our chloroplast, hence this is our location. Now, because we've got this 6-carbon molecule, it's actually a very unstable molecule, and unstable things break down pretty quickly. So what actually happens, that 6-carbon molecule, whose name we don't care about at A-level, is going to break down to form 2 molecules of a chemical called GP. Again, GP works. If you want to know its full name, it's glycerate 3-phosphate. Now, this is a 3-carbon compound, and hence we get 2 of them, because if we're starting off with this unstable 6-carbon molecule, it's going to break down to form 2 3-carbon molecules of GP. Now, that process that we've talked about so far is what we refer to as carboxylation. And the reason that we call this carboxylation is because the carbon dioxide has been fixed and we have now formed our GP. The next thing that's going to happen is we're going to have our 2 GP molecules, which remember are 3-carbon compounds, and they are going to form 2 molecules of this stuff called triose phosphate, or TP for short. Now, to change our GP to TP, what we need to happen first of all is we need to reduce the GP. And to reduce it, we're going to add hydrogen. The hydrogen itself actually comes from one of those products of the light dependent stage. Now, at the end of the light dependent stage, we generated reduced NADP, and this is then going to be fed into the light independent stage here. So our reduced NADP is then going to give up that hydrogen and become reoxidized to form NADP. So what we actually see is this is going to happen with 2 of them. So we've got 2 reduced to form 2 NADP. And at the same time, we're going to see ATP being converted into ADP and inorganic phosphate. Now, what's actually happening here is we're utilizing the energy from that ATP to actually bring this change about. So what we can see at this stage, 2 GP molecules reduced by the addition of hydrogen from the reduced NADP from the light dependent stage. That then regenerates the NADP, and we're converting ATP into ADP and inorganic phosphate. End result, we get 2 molecules of TP, triose phosphate. One thing that we do need to remember is that 10 out of every 12 TP molecules that we generate are actually going to be used to reform the RuBP, the ribulose bisphosphate. Now, that occurs through further phosphorylation with the ATP there. The other 2 out of every 12 TP are the product. So we're not having a massively efficient process here at making TP, because obviously 10 out of every 12 are just going back to reform the RuBP. So what we actually find is, in order to make glucose, which is obviously the other end product of photosynthesis, we're going to need to go around the Calvin cycle 6 times to form the 2 molecules of TP that we are then going to use to make the glucose there. This is the full thing then. Okay, so we've gone through the individual little bits. Now let's look at it as a big overview diagram. So here's our starting point with our RUBP that we talked about earlier. So we start off with our 5-carbon RUBP. That's then going to combine with the 1 carbon from carbon dioxide in the presence of our little enzyme rubisco, forms that unstable 6-carbon molecule that immediately breaks down to form 2 molecules of GP, 3 carbons. The GP then is converted into TP, with obviously the reduced NADP being reoxidized to NADP, and the ATP being converted into ADP and inorganic phosphate. From that, the TP will mainly be going back to reform the RUBP, and some will go on to make our final TP product. So what we can actually see is we basically have 3 stages we can break this down into. Stage 1, which is from our RUBP to the GP, that is our carboxylation. So we've added the carbon dioxide to form this organic molecule. Stage 2 is the reduction. Then stage 3, the regeneration of the RUBP. As we said at the start of this video, the Calvin cycle is in the light independent stage. It doesn't need light directly to function. However, without light, this all grinds to a halt very quickly, for the simple fact that the Calvin cycle requires ATP and reduced NADP, which are products of the light dependent stage. So if we don't have light, we won't have the light dependent stage, we won't be forming ATP and reduced NADP, which means that we won't then be able to run the Calvin cycle. We'll run out of those 2 things very quickly. The other thing to bear in mind, we've mentioned this enzyme rubisco, and the optimum for that enzyme is pH 8. Now, what we do hopefully remember from the last video is that at one point we were talking about hydrogen ions being pumped into those thylakoid spaces. Now, because we're pumping hydrogen ions from the stroma into the thylakoid spaces, what we're actually doing is removing hydrogen ions from the stroma, and hydrogen ion concentration is what gives us our pH at the end of the day. So if we're reducing the concentration of hydrogen ions, then we're making the pH become more alkali. So we're moving it towards that pH 8 because there's a lower concentration of hydrogen ions present, which means we've shifted the optimum pH closer to what it should be for our rubisco. The last thing we really need to know then is the second spec statement we're looking at. What do we use this triose phosphate for, or the TP? Now, triose phosphate itself is used to synthesize a whole range of different organic compounds. So we will use the triose phosphate to synthesize amino acids, we'll use it to synthesize fatty acids and glycerol, and those will then obviously go on to combine to form our lipids, back to our biomolecules work. We will also be able to use our TP to synthesize carbohydrate in the form of these hexose sugars, like glucose. And we will also use TP to reform our ribulose bisphosphate, the RUBP. So make sure you know at least 3 of those from our list, because that way, whatever you're asked to do, you should be able to answer it. Make sure you remember reform RUBP without a shadow of a doubt, and then 2 things that we can also synthesize from TP, and you'll be absolutely fine on those questions. Don't forget to subscribe to the channel so that you can see when we next upload another video, and of course head on over to the A-Level Biology website where you'll be able to find a range of other resources to help you with your A-Level biology studies.